potassium will be pumped outside to the extracellular fluid, and 3 sodium and 2 potassium will be pumped back. So since it is 3 sodium, 2 potassium, so you lack 1. So you will have a negativity inside the cell. Okay? So with that negativity, when the sodium will, when 1 sodium will come in, then that will make the inside of the cell positive, right? And so that is already that can create an action potential. So how does this action potential be propagated? Going towards the, for example, you're from the neuron of the cell, and then you go towards the muscle. The nerve goes toward the muscle. So how will action potential be propagated? So Ian, so for every, since we are using the nodes of Ranvier, saltatory conduction, the nerve is using saltatory conduction. So for every nodes of Ranvier, that is where the action potential is being produced. So from one node of Ranvier to the next. So the action potential will be propagated very fast. So continues, the sodium will come in, and so on the next node of Ranvier, the positivity, you know, the positive inside the cell will create a current. And so this going inside the cell of the sodium is continuous. So, but it will jump from saltatory conduction from one node of Ranvier to the next. So that is how the action potential is being propagated. So it will create a current and going to the muscle. So then it will reach up to the neuromuscular junction, okay? So this morning, we will discuss about muscle physiology. This one, you have your skeletal muscle, cardiac muscle, and smooth muscle. This is your skeletal muscle, cardiac muscle, and smooth muscle. And so, what do you notice? There are striations in the skeletal muscle and in the cardiac muscle, but the striation is not much as in the skeletal muscle. And here in the smooth muscle, there are no striations, no? Because the the cardiac muscle has intercalated discs, while the striations of the skeletal muscle is due to the sarcomere, okay? The thick and thin filaments. Muscle fiber is a single skeletal muscle cell. This is your muscle fiber. Can you hear me?
Yes,
Yes, Doc.
The muscle is attached to the bone by a tendon, and this is wrapped by a connective tissue. And each muscle cell is wrapped by a connective tissue also. And so you have several muscle fiber that is bound together by connective tissue. So these are attached to the bone by tendons. Now these striations is due to the thick and thin filaments. The sarcomere is the functional unit of the muscle. That is from Z line to Z line. This light portion to the next lighter portion. The center is made up of, excuse me, the thick filaments. The thick filaments is composed of myosin, entirely protein, myosin. The thin filaments is composed of proteins, your actin, troponin, and tropomyosin. The thick filaments are in the middle of each sarcomere. So it's here, okay? And each sarcomere has two sets of thin filaments at the sides. Two sets, and they are bound to the Z line, one on each side. One end of each thin filament is anchored to an interconnecting protein called your Z line, while the other end overlaps a part of the thick filament, okay? And that portion where the thin filaments end in the middle of the sarcomere will form your H zone. But at the center of the H zone is your M line. M line. Two successive Z lines are the boundaries of a sarcomere, this one, okay? Two successive Z lines. So there will be another Z line here. Are the boundaries of a sarcomere. A light band, this one, will form the I band. It's composed of the thin filaments on either side of the Z line. The thin filament, before it reaches the Z line, will form your I band. So there are two I bands for each sarcomere, okay? So you'll have two I bands. There is an I band before every Z line. Since there are two Z lines for each sarcomere, then you have two I bands. These are the thin filaments where no thick filaments are located. So in the center, before at the termination of the thin filaments, at the center of the sarcomere, that space will form the H zone. It corresponds to the space between the two opposing ends of the two sets of the thin filaments. Okay? The M line is a narrow dark band at the center of the H zone. These are proteins that link together the central area of the adjacent thick filaments. Excuse me siya. I-admit na naman itong mga tao na to. Kasi nag-interfere siya sa slides ko. Paki-admit siya. Thank you. Okay. Clear so far? That is the general formation of your sarcomere. So your M line is here, the H zone. M line is located at the center of the H zone. The elastic protein, titin, will extend from the Z line to the M line. From the Z line to the M line, going through the thick filament. This acts to maintain the correct alignment of the thick filaments in the middle of each sarcomere. Sort of an anchor. Huh? Yeah. And the thick this is your thick filament, okay? And this is your cross bridge. And this is your thin filament. It is composed of your actin, tropomyosin, and troponin. Okay. That is your titin. Contraction of your skeletal muscle. Because it is the thin filament from each end of the sarcomere that slide towards the center during isotonic contraction. The sarcomere, this one will slide, okay? This one will slide towards the center, your thin filament. So what happens to the sarcomere? It will shorten. So your sarcomere will shorten and the I band is narrowed. This I band is narrowed, okay? The H zone is reduced. Notice the Z lines are moved closer. Your Z lines, they are moved closer. The A band, this is your A band. It is composed of your thick filament. this will remain unchanged because the thick filament will not slide. It is only your thin filament. The thick and thin filaments do not change their length. So if I will say in the question, one of the following will not occur. The following will happen when thin filaments slide towards the center during isotonic contraction. A, the sarcomere shortens. B, the I band is narrowed. C, the H zone is reduced. D, the A band. E, the Z lines are moved closer. One of the following does not occur when the thin filament slides toward the center of the sarcomere during isotonic contraction. So it is always like that. The question is during exams, the questions are multiple choice questions. Select the best answer. So what is your best answer there? The sarcomere shortens, the I band is narrowed. Letter C, the H zone is reduced. Letter D, the A band shortens. Letter E, A, B, C, D. D,
D.
The A band is shortened. Letter E, the Z lines are moved closer. So it's like that. That is an example of the questions that will be given to you. They are all multiple choice questions. Okay, even if it is a problem, you can solve it through your scratch paper. But the question is still A, B, C, D. You still have to choose A, B, C, D. But, oh, I forgot. Nowadays, it's no longer A, B, C, D, E. It's only 4 choices, A, B, C, D. That is the new format of the questions. A, B, C, D from the APMC. So, there is no longer letter E, only up to letter D. Okay, so you take note of that. That is, so during the process of your studies, when you are reading, more or less you evaluate what could be the possible questions that can be made out of this. Okay, so that is already one. Okay. Now, these observations led to the conception of the sliding filament mechanism of contraction. Take note, it is only the thin filament that slides. The thick filament will stay put. So the A band will remain the same. It will not shorten. Okay? Yeah. So your thick filaments is found in the A band, okay? I've already mentioned that. It contains myosin. Myosin has heavy and light chains. This one, the light chains and the heavy chains. Each has 2 heads attached to a single tail. These are the heads of the thick filament. The myosin head have two binding sites. The one that binds to the actin and the ATP binding site below. The ATP binding site serves also as your ATPase that hydrolyzes the ATP, using its energy for contraction, because you spend energy to contract the muscle. And the myosin heads, they're involved in cross-bridge formation. These are the ones that will attach to the thin filament. This is your troponin. It covers your actin. And then you have your the tropomyosin covers the actin. And then you have your troponin. That is, you have three kinds of troponin here. We'll try to see. The thin filament is formed by the aggregation of actin molecules, globular actin or G-actin, into a two-stranded helical filament called F-actin or the filamentous actin. Nebulin is elongated cytoskeletal protein that extends along the length of the thin filament and may regulate the length of the thin filament. The nebulin is more or less like your titin in the thick filament, okay? Now your tropomyosin extend over the length of the entire actin filament, and they cover myosin binding sites on the actin molecules. Each tropomyosin extends over around 7 actin molecules and cover them. The two filaments, the thin filaments, they are anchored at the Z lines and present in the I bands. They contain actin, tropomyosin, and troponin. One of the following is not a component of tropomyosin. Troponin. Troponin A, troponin T, B, troponin I, C, troponin C, D, troponin A. Sort of like that, okay? So if you already know, you have memorized the 3 types of troponin, then of course it is A. There is no troponin A, okay? It's troponin T. Troponin T, this attaches the troponin complex to your tropomyosin. And troponin I inhibits the interaction between the actin and the myosin. The troponin C, this is the calcium binding protein, where the calcium will bind. Once the calcium will bind to the troponin, this will activate troponin T, and once this attaches to the tropomyosin, the troponin T, this will uncover the actin binding sites, okay? So that the head of myosin can attach, so that the actin can attach to the head of the myosin binding sites, okay? So remember that troponin T, troponin I, and troponin C, okay? The thin filament contains actin, troponin, and tropomyosin. It is a complex of protein, so that your troponin is composed of three types: T, I, C, thick. Okay. Now, how does contraction take place? First, the calcium binds to troponin. Where does the calcium come from? From your sarcoplasmic reticulum in the cell, muscle cell. So your calcium binds to your troponin, okay? This is a resting muscle. So this is how your thick filament appears. So the head, the cross bridge is standing already, okay? And then there is the binding site exposed to the actin. Binding site to actin is exposed. So once calcium binds to troponin, the tropomyosin will uncover the actin. Uncovers the actin site, and once it is exposed, it will attach to the head of the cross bridge, okay, of the myosin. Then the cross bridge will tilt towards the center of the sarcomere. This is your cross bridge will tilt towards the center of the sarcomere, pulling the actin along, because the actin has already attached to the actin binding sites. The ATP breaks the actin-myosin bond. So the binding of the cross bridge, the binding of actin to the cross bridge will spend one phosphate. So your ADP will be phosphorylated to ATP. And then this is spent, and your ATP will go back to its original ADP form, adenosine diphosphate. Okay. Upon binding to myosin, this will slide. Your actin is brought along when your head, cross bridge head, will tilt towards the center. Okay. So your ADP, this has already released your phosphate because it has been used already. So your ATP will go back into ADP. And your ATP, and this is phosphorylated. The cross bridge will move, yes. So ADP plus phosphate. And then this one, there is another ATP binds to myosin, causing cross bridge to detach. This will use again ADP, and then plus phosphate. This will form ATP. Yeah. The ADP, when it is not yet sliding, and the actin will attach to the binding site of the cross bridge. So ADP plus phosphate will form your ATP. And this is the one, your ATP, it is the one that causes the cross bridge to detach from your actin binding sites. And so it will spend the ATP there, causing cross bridge to detach. Once ATP is spent, so hydrolysis of ATP energizes the cross bridge. And so it will detach because it has energy. Then your phosphate will go back to your ADP form. Okay? So it will go back to the beginning. If there is no ATP formed after death, so it will remain in its attached form, okay? The cross bridge could no longer detach because there is no more supply of ATP because death has ensued. Okay, so it will remain there attached to the actin, and that is, so it will harden. That is your rigor mortis during death. This is the explanation why the muscle will harden after an individual dies, okay? It will go into rigor mortis. The cross bridge will remain attached to the actin, okay? Because there is no more ATP. So the energized cross bridge springs back to original position, and it will bind again to the next actin site, okay? So, and this will repeat 12 times. So your actin, your thin filament is moved towards the center, but your myosin will stay put. It is only the cross bridges that will do the action. The binding of cross bridge, ATP to the cross bridge will be repeated 12 times before contraction is elicited. Now, where do we get our calcium? From the cross bridge, from the sarcoplasmic reticulum. This is how calcium is being taken, derived. Remember, in your muscle has sarcoplasmic reticulum. And your muscle plasma membrane has T tubules that invaginates towards the interior of the muscle cell. It has T tubules. The T tubules, the action potential will pass through the T tubules.